Hi, good evening, everyone. So we will start with week 10 tutorial session. Okay, we'll summarize the lecture content and then look at few sample questions. My voice is a little hoarse this week, so just bear with me. Okay, I will start sharing the screen. So this week again, as part of how are we going to really characterize the product that we have purified. One, we looked very extensively into gel filtration chromatography or size exclusion chromatography to determine the native molecular weight, then electrophoresis and mass spectrometry. So now we will look at in this tutorial content, tutorial session, we will look at I like spectroscopic methods and sequencing methods to uh, determine or characterize the product's concentration as well as the integrity of the product, as in the purity of the product. Okay. So now we'll start with sequencing, different types of sequencing of protein molecules. Okay. So when we talk about protein sequencing, like we looked at DNA sequencing, right? Where we use dideoxynucleotides to stop the chain extension. And then wherever the like dideoxy, like DDATP, DDTTP, DDGTP, all these molecules are there, the reaction stops there because the chain can't be extended from there because of the lack of uh, oxygen molecule. Okay. So that's how we are able to stop the reaction, and then we run it on like really large gels along different lanes corresponding to A, T, G, C, and then accordingly we will able to match the we will able to annotate the different bases or nucleotides in the digested fragment. Okay. So similarly, protein sequencing is also really an essential workflow in biology and medicine. It enables the analysis of amino acid sequences, which confirms along for which one does for confirmation along with the protein structure and function. To get an idea about the protein structure and function, one can even identify new protein biomarkers and drug targets. One can even construct the phylogenetic trees and to understand the evolutionary relationships, and one can even identify orthologous and paralogous proteins, and overall this helps in extrapolating the functions of unknown proteins, facilitating the cDNA library screening and supporting the protein engineering studies. Now when we talk about orthologous and paralogous genes, okay, so in general, what are orthologous genes? These are genes from different species which have been separated by speciation. Okay. Whereas paralogous genes are found within a single species, but it has duplicated eventually. Okay. like like due to gene duplication, now they are part of different uh, gene groups. Okay. And these orthologous genes carry out the same function as that of as that carried out by the ancestral gene, but paralogous genes can have actually different functions other than the function that the like from which these have eventually delineated. So orthologous genes are due to speciation event, and paralogous genes arise due to uh, gene duplication event. So orthologous genes are present in different species Okay, and paralogous genes are present in the in a single species, and this is what is similar to the thing here, like excuse- orthologous genes are similar to ancestral genes, and paralogous genes are combinations of ancestral as well as, like, it can be combinations of ancestral and new genes. So when we talk about, excuse me, suppose there's a pair of homologs or homologous genes. So frog has alpha form, chicken has alpha form, and mouse has alpha form. Mhm. So these three have chain of a particular gene, suppose, and another gene encodes the beta chain. Okay. So in frog, chicken, mouse, all the alpha chains are encoded, and in the same organisms, all the beta chains are encoded. So what we will say is frog alpha, chicken alpha, and mouse alpha are actually orthologs of each other. Okay, these genes encoding the alpha chain peptide, suppose if we are talking about a peptide, then these frog alpha, chicken alpha, and mouse alpha are orthologs of each other. Similarly, the mouse beta, chicken beta, and frog beta are orthologs of each other. But mouse alpha and mouse beta, what actually had happened, this might have been the very early ancestral gene of interest that we are talking about, and then it duplicated and bifurcated into alpha chain gene and beta chain gene. Now the mouse alpha and mouse beta will be called as paralogs of each other. Okay, so that is what this was being told here, like paralogous genes are within the same species, like within the mouse species only, the alpha and beta chain genes which had duplicated earlier from an ancestral gene are paralogs of each other. However, in orthologs, it is in different species when the same one particular gene is encoded, like encodes the same or like similar, not same, similar, suppose peptide, protein peptide, or similar peptide chain, like here alpha chain, but in different species. So then these are orthologs of each other. Okay. Similarly, orthologous and paralogous proteins, as a general rule, orthologous proteins tend to perform the same function in different species. Okay, while paralogous proteins tend to have diversified somewhat in function. How? Via duplication. So, duplication is a very common way in which evolution gives proteins the freedom to develop new functions. For example, if we are talking about chymotrypsin, serine proteases are orthologous to each other. Chymotrypsin enzyme and serine proteases are orthologous to each other, and these enzymes or proteases retain not only the same general function, proteolysis, which is proteolysis using a catalytic triad including a serine, but these proteases also have the same substrate specificity. That means it cleaves at positions uh, following an aromatic side chain. Okay, or cleaves at positions where amino acids having like aromatic amino acids are present. After an aromatic amino acid, these two uh, uh, enzymes cuts at those specific positions. Okay, so but so these are orthologous to each other because they have retained the same general function as well as the same substrate specificity. But chymotrypsins are paralogous to the trypsins and the elastases. These are again enzymes. Okay. Now, when we talk about chymotrypsins, trypsins, and elastases, now all these proteins share the same general serine protease function, but these have evolved different substrate specificities. So these proteins also have paralogs which have lost all protease activity. Okay, so in the previous slide, we looked at orthologous versus paralogous genes, and this is the same analogy can be extended to orthologous and paralogous proteins. So now, basically, what is protein sequencing? It is the process of examining the covalent structure and amino acid sequence of a fully developed polypeptide, which is considered a component of post-translational modifications. And the structure of a protein is intricately linked to the sequence of amino acids in that protein. The sequence actually dictates the structure, and the structure eventually dictates the function. Okay. Now, what are the different advantages or benefits? Why do we really need to do protein sequencing? We looked at it in a brief like overall brief view in the first slide. So one is decoding genetic information. So at its core, protein sequencing serves as a bridge between the genetic information encoded in DNA and the functional proteins that carry out essential cellular processes. Then one can unveil the protein function. Definitely, the primary structure of a protein is dictated, I mean, the structure or the sequence of the amino acids dictates the function of the protein, and each protein's unique sequence dictates how it folds into its three-dimensional structure because of the interaction, like intra-chain as well as interchain interactions. Okay, and consequently how it interacts with other molecules within a cell. Understanding the sequence, amino acid sequence, is crucial for understanding the roles proteins play in biological systems. Okay, and it can eventually have implications in biotech. What happens is it's a fundamental step in the design and production of different biopharmaceuticals, enzymes, and genetically engineered proteins. By precisely sequencing target proteins, researchers can engineer these molecules for specific functions or therapeutic applications. It can be used for personalized medicine as well. Uh, so it enables, like protein sequencing enables identifying the genetic mutations and variations associated with diseases, which eventually allows for the development of tailored treatments and therapies for individuals. Definitely one will get structural biology insights. Okay, so before researchers can actually look into the 3D structures of proteins, basically the folded tertiary and quaternary structures, so they must determine the primary structure, which is the basic amino acid sequence through protein sequencing. It is essential, it is actually very crucial for structural biologists seeking to understand how a protein's shape relates to its function and interactions. Okay. And in the broader context of proteomics, the study of all proteins within a biological system, protein sequencing is foundational. It facilitates the identification of proteins, what are the modifications the residues are going through, and their interactions, eventually helping researchers unravel complex cellular processes and signaling pathways. So, one way to do uh, protein sequencing is breaking the disulfide bonds. Okay, so the disulfide linkage actually interferes with the complete sequencing procedure because it doesn't allow the release of cleaved amino acid from the peptide chain. So there are basically two approaches to disrupt the disulfide linkage. First approach is protein is oxidized here with the performic acid here, with the performic acid to produce two cystic acid residues here. Or one can eventually use these reducing agents like DTT. Here, it is DTT is used. These reducing agents like DTT or BME is used to form two cysteine. Like this is the disulfide linked protein, and then it is DTT is added or beta mercaptoethanol is added. Actually DTT and BME both are reducing agents. It's fine. But one thing is there, like DTT is less stable, comparatively less stable to BME. Okay. So what usually is done is BME can be like it's stored at room temperature And but DTT needs to be always stored at, like, for short term use, maybe within a week or so, or actually it's always good to really freeze it and store it at -20 and just thaw it before use. Okay, because it's relatively unstable. I'm talking about DTT here. Hmm. So DTT, whenever while preparing buffers or for any reaction, wherever it's needed, DTT should be thawed before use and then added immediately before final use. Okay, but BMA comparative, BME should also, being the reducing agent, it should also be added before use, but it's comparatively more stable than DTT. Okay, but anyways, depending on the availability as well as one like researchers preference as well as the type of reaction they want to try out, one can eventually use DTT or BME, and accordingly, like, just one needs to keep in mind the storage conditions and when to really add the thing. Okay. So yeah, but yeah, we were talking about sequencing, right? So first the protein has to be reduced by DTT or BME, some kind of reducing agent, to form two cysteines, two cysteine residues, followed by treatment with iodoacetate. So iodoacetate actually leads to carboxymethylation here. Okay. So this is what is happening here. This is the carboxymethylation happening, and what happens is, if the carboxymethylation takes place, then this S, these sulfur residues won't really be able to uh, again uh, be able to lose the H atoms here to form a disulfide linkage. Okay, this carboxymethylation due to iodoacetate addition helps in preventing the formation of, like, the reversible disulfide linkage formation is basically stopped. So iodoacetate is commonly used to alkylate free thiol groups during the reduction of disulfide bonds between cysteine residues. When disulfide bonds are reduced, free thiol groups or SH groups are generated. Okay, like in this reaction here. Okay, and iodoacetate is then added, which reacts with these SH free SH groups to form carboxymethylated cysteine residues, effectively preventing the reformation of disulfide bonds. This is actually crucial in experiments where maintaining the reduced state of cysteine residues is is an absolute necessity for further analysis or protein characterization. Next is, you again, how you are doing DNA sequencing. You basically want to sequence a particular stretch of DNA having, let's suppose, 200 bases long. The DNA sequence is 200 bases long. You chop it off into multiple fragments, right? Similarly, you have to cleave a long polypeptide chain, like long amino acid sequences. And how do you really cleave that? For DNA fragment, you used we use DNases. Okay, like, mainly nucleases, different kinds of nucleases. And similarly, for a polypeptide chain, it will be proteases, and so, along with a mixture of certain chemical agents which target proteins at specific sites, amino acid sequences, and then cleave after a particular amino acid. So these are few examples. Suppose this trypsin enzyme cleaves after uh, whenever after lysine or arginine residues are there. Chymotrypsin cleaves after phenyl, alanine, tryptophan, and tyrosine residues, basically aromatic amino acid residues. Then pepsin is there, which actually can cleave after basically cut the polypeptide chain after a leucine residue, phenylalanine residue, tryptophan residue, or tyrosine residue. And there's this reagent, cyanogen bromide, which cleaves after methionine. Okay. So protein sequencing can be categorized into three main methods. One is to investigate the N-terminal part, the C-terminal part, and or you eventually, what I was talking about, you break down the polypeptide into multiple short peptide fragments. Okay. So when we talk about N-terminal sequencing, So at the N terminal, you have free NH2 group, right, of a polypeptide chain. Now, the N-terminal amino acid analysis is a three-step process basically. What happens is, the first is you label the terminal amino group, NH2 group, with compounds like DFNB and dancyl chloride here. Okay, this is the N-terminal portion of the long peptide chain. If there's free NH2 group, there is this reagent DNFMB. Basically, it has like two NO2 groups along with the fluorine attached to the benzene ring. And in most of the cases, these reagents can also label free amino group present on basic amino acids like lysine and arginine. Right? In the side chain of lysine and arginine, you have multiple NH2 groups as well. So those can also be labeled by these reagents like DNFB reagent. So what happens here is, uh, dinitrofluorobenzene or DNFB reacts with the free amine group here, NH2 group, to form dinitrophenyl amino acid complex. This one. Dinitro because there are two NO2 groups attached to uh, this benzene ring, and then this, after losing this H from here and fluorine atom from here, basically this link, like this adduct is formed. And then finally, uh, hydrolysis takes place where you have mainly acid hydrolysis is taking place of this particular dinitrophenyl amino acid complex, and it breaks down the peptide bond to release dinitrophenyl amino acid complex in solution, and eventually all these separated products can be analyzed on the chromatographic column or on a like TLC plate, basically via chromatography. One can eventually separate out these different spots. Okay. Then there's something called Edman degradation, which is used for purifying proteins by systematically eliminating individual amino acid residues from the peptide's N-terminal end. Okay. Amino end. Now, this was actually devised by a person named Per Edman. He devised a solution to the problem of potential protein damage during hydrolysis, right? So instead of using harsh conditions, what what Edman did was introduced a novel labeling and cleaving approach. First, you label the N-terminal part of the amino acid, then eventually cleave due to like at a particular pH, at a particular buffering condition, and then by adding phenyl isothiocyanate, a phenyl thiocarbamoyl derivative can be formed with the N-terminal part here. And then cleavage of N-terminal occurs under mildly acidic conditions, okay, resulting in a cyclic compound here, which is PTH, phenyl thiohydantoin amino acid, or PTH. Okay, under mildly acidic condition, this whole complex gets hydrolyzed and forms this cyclic PTH compound. And this complete process preserves the protein integrity, leaving behind two peptide sequence components. Okay. So this procedure can be repeated for each residue, allowing for the sequential separation of individual residues without compromising the overall protein structure. All right. So Edman degradation is actually useful because it is not damaging the protein. We are eventually removing one residue after the other. And this allows sequencing of the protein to be done in a comparatively lesser amount of time. And it is done best if the composition of the amino acid is known, definitely. And to determine the composition of the amino acid, the peptide needs to be actually hydrolyzed. And the protein has to be denatured by heating it and adding HCl for a long time. Then what happens is individual amino acids eventually separate, and then amino acids can also be separated by ion exchange chromatography, because different amino acids can have, like what we were looking at, it has different owing to the type of side chain that one has, what kind of side chain, whether it's a polar group, whether it's basic group, acidic group, whatever side chain is there, based on that, one can have a, like the amino acids can eventually take up a net charge on it based on what is the pH of the surrounding medium, right? So eventually, so we can play around with the pH of the medium and separate it by ion exchange chromatography. Then after separating out, these are then dyed with ninhydrin solution, ninhydrin reagent, and what is the amount of amino acid that can be determined by the amount of optical absorbance. So basically the solution's color changes and how much is the concentration of the amino acid. Uh, I mean, the concentration of the amino acid in that particular solution can be detected. This way, the composition but not the sequence can be determined. Okay, which is coming after what we can't really know, but we can eventually know that okay, how many cysteines are there, how many serines are there, kind of like that. Because okay, now when we talk about ninhydrin test, uh, these are like I think we used to learn in high school chemistry, right? Practicals that we used to check whether a given sample has amines or alpha amino acid. Okay. Now, ninhydrin is basically again a reagent which is added to a test solution of the sample in question, and the whatever color develops, it indicates the presence of ammonia, primary or secondary amines or amino acids in the analyte. Okay. So, this is the amino acid in question. So, amino group belonging to free amino acid, here NH2, undergoes a chemical reaction with ninhydrin. Ninhydrin basically behaves as an oxidizing agent. Okay, that means it itself is getting reduced. So, when exposed to ninhydrin, the amino acid in question undergoes oxidative deamination, resulting in the release of CO2, NH3, CO2, NH3, and an aldehyde. Here, aldehyde along with hydrinantine, or which is eventually the like a reduced form of ninhydrin. And then the NH3 that has eventually come out goes on to react with another ninhydrin molecule to form Ruhmann's complex. Okay, this complex is responsible for the development of the deep blue color here. And when the analyte contains amino acids like proline, a yellow colored complex is formed. And when asparagine is used, the color of the resulting complex is brown. Okay. So larger proteins cannot be sequenced by Edman sequencing because of the less than perfect efficiency of the method. Edman is what one by one individual amino acids we are trying to sequence. Yeah. So because it's too large, too large a polypeptide chain, this eventually compromises the efficiency of the method. And a strategy called divide and conquer. Basically, you have to digest it like a very large polypeptide chain can be eventually reduced to multiple smaller number of peptides, which are eventually which can with the larger protein can be cleaved into smaller practical like amino acid sequence of practical lengths, and this is done by using certain chemicals or enzymes which can cleave the protein at specific amino acid residues. And the separated peptides can be isolated by chromatography, and then these can be sequenced by Edman method because of their smaller size. Now, in order to put together all the sequences of the different peptides, a method of overlapping peptides is used. If you remember the tutorial from, or in general, the concept from DNA sequencing, that's how whenever there was overlap in the matches, that's how one annotated the particular nucleotide in that position corresponding to in which well things are turning up. Okay, so in order to put together all the sequences of the different peptides, a method of overlapping peptides is used. The strategy of divide and conquer is basically protease digestion. followed by Edman sequencing is used again a second time, but using a different enzyme or chemical to cleave it into different residues, right? I mean, it would have been good if I showed it here. So basically, suppose chromatography, okay, this is a peptide sequence. Uh, I'm just giving a vague example, and you used enzyme A to cleave at positions, which only cleaves positions between O and like after O. Okay, after O residue, there's no amino acid residue called O, but this is just a way example what I'm talking about. It will only, like, enzyme A will cleave only after O residue, whereas another enzyme, so you will eventually have there are because there are two O's here, so you will eventually have three peptide fragments, right, of this complete peptide sequence. Of the larger peptide sequence, you will eventually have, it will eventually cut off here, here. So eventually you have CHRO sequence, then there will be MATO sequence, and then there will be a graphy sequence. Okay. So similarly, if we now use another enzyme B, suppose another protease B, which only cleaves after A residue. Okay, so then again it will also give off three peptide sequences, but of different composition, right? So it will have chroma, then TOGRA, then PHY. Okay, so now when we eventually see how these things are lying up, so there will be overlying residues, sorry, I mean overlapping residues, terminal residues. Okay, either it can be a stretch or one residue, whatever. So those can be eventually looked at to find the complete sequence of the peptide chain. Okay. Um, so what is happening here? You first um, digest it with a particular protease having a different substrate specificity, or having a different cleavage site as compared to another protease which has a totally different um, cleavage site, and eventually you generate different types of fragments from different resulting from different different proteases that have been used, and then eventually you can use the overlapping peptide sequences to finally get to the original original amino acid sequence. So by comparing these two sequences and examining for any overlap between the two, suppose if we are using two proteases having different cleavage sites, what I was talking here, uh, the sequence can be known for the original protein. Okay. So, for example, trypsin can be used on the initial peptide to cleave it at the carboxyl side of arginine and lysine residues. Okay. And uh, using trypsin to cleave the protein and sequencing them individually with Edman degradation will yield many different individual results. Although the sequence of each individual cleaved amino acid segment is known, the order is completely jumbled, right? Now, another protease, chymotrypsin, which cleaves on the carboxyl side of aromatic residues and some other not like bulky non-polar residues. One can even use chymotrypsin and then sequence the chymotrypsin digested segments. So there will be overlap with the chymotrypsin digested segments as and the trypsin digested segments. And then the overlapping residues can be checked properly to find the original sequence of the initial protein. But this method is limited in analyzing very large sized proteins, okay, more than 100 amino acids because of secondary hydrogen bond interference. And there might be other weak intermolecular bonding like hydrophobic interactions, which cannot really be properly predicted. Okay, so only the actually linear sequence of protein can be properly predicted, assuming the sequence is, it's scalable. Basically, it's small enough and one can eventually sequence like within 100 amino acids. It's a pretty good number. Otherwise, like really large amino acids, amino acid sequences is tricky. Sequencing really large protein sequences is actually a little tricky. Mm. Next, one can even look at the C-terminal residues, but not many methods are developed for C-terminal amino acids analysis. And the most common method to do is to treat the protein with carboxypeptidase to release the C-terminal amino acid and test the solution in a time-dependent manner. Then this is ordering the peptide fragments. The usage of different protein cleavage reagent produces overlapping amino acid stretches, the same thing that we were talking about. And these stretches can be used to put the whole sequence. One can also locate the disulfide bonds. Protein cleavage by trypsin is performed with or without breaking disulfide linkage. And then one does the amino acid sequence analysis of the fragments to provide the site of disulfide bond. The presence of one disulfide will reduce two peptide fragments and will appear as one large peptide fragment. Okay. And then eventually we have mass spec, which is used to determine the protein's mass and structure. What happens is you ionize the peptide sequence and accelerate it. Uh, so ionize and then accelerate these ionized particles through a mass analyzer, and eventually you detect it on a detector. And so basically these uh, ions, whatever charged particles are there, due to their different mass and size, they eventually hit the detector at different time points. So this is eventually detected by the detector based on the deflection, and this particular deflection is actually proportional to the m by z ratio. So sample introduction methods. So mass spectrometry can have different types based on like how you are introducing the sample into inside the mass analyzer. It can be diffusion, liquid injection, laser desorption for large proteins. Okay. So there is something called LCMS, which is a very common technique. It employs electrospray ionization for a reliable interface. It's versatile for various biological molecules, and then tandem mass spec and stable isotopes can enhance sensitivity and accuracy. Despite the need for the method optimization to reduce ion suppression, fast scanning enables multiplexing, measuring numerous compounds in one run. As instruments become more affordable, LCMS is gaining importance in clinical biochemistry, competing with traditional methods like liquid chromatography and immunoassays. Then there is something called quadrupole mass analyzers, which select the ions, whatever charged particles one has generated, based on varying m by z ratios, and employ collision induced dissociation to fragment the selected ions in the ion trap, allowing the determination of peptide or protein sequences. This technique typically involves two mass analyzers separated by a collision cell, but it can also be accomplished in a single mass analyzer. There's a like basically the setup differs. Okay. And finally, we have spectroscopy, which is a technique where we study the interaction between the matter and electromagnetic radiation. Whenever the matter is exposed to some kind of electromagnetic radiation is actually a spectrum of different kind of uh, waves uh, at different wavelengths. Okay. And the matter in question can be ions, molecules, or atoms. So, spectroscopic techniques employ the fact that light tends to interact with matter and exhibit some features of a sample, which helps us to know much about its consistency or structure. So, the nature of the interactions in spectroscopic techniques between radiation and matter may include. So, just. So, yeah. So this technique employs uh, light. This technique employs the fact or uh, that light tends to interact like any light, any matter exposed to certain light interacts and exhibits some features. So what can be the different interactions in spectroscopic techniques? It can be absorption, emission, or scattering. Now, one is qualitative spectroscopy, where we study the radiation absorbed or emitted by an atom or molecule of the matter in question, or emitted by the matter in question, which will give information about its identity. And one is quantitative spectroscopy, which will give information about the number of absorbing or emitting atoms or molecules by measuring the total amount of radiation. So in general, uh, electromagnetic radiation have, you know, mutually perpendicular electric and magnetic field, uh, which are actually perpendicular to the direction of the propagation of the wave. Okay, and this is, the red one is the electric field. Okay, suppose uh, suppose if we say that this is the z axis, this is x and y. So if the direction of wave propagation is y axis, then the magnetic field or b vector is along the x axis, and the e vector is along the z axis. Okay, just this might not be the case, it can be a different scenario altogether. Just uh, just uh, for an example, now if we are talking about the electromagnetic spectrum, then it has uh, radio waves, microwaves, infrared waves, UV rays, X rays, and gamma rays. Gamma rays having the highest frequency, and but the least wavelength of among them all, and radio waves having the highest wavelength. So uh, basically these are inversely proportional, right? The wavelength and frequency of the light wave or wave in question. So, and also in between here we have the visible spectrum lying somewhere between 400 to 750 nanometers wavelength range. And based on this, there are three principal actually in there are three principal branches of spectroscopy. One is absorption spectroscopy, which uses the range in which a material absorbs the electromagnetic spectrum. It basically involves the atomic absorption spectroscopy and different molecular techniques in that area, such as infrared spectroscopy. In the radio region, there's NMR spectroscopy. And then there's another called emission spectroscopy, which uses the electromagnetic spectrum range in which a material actually radiates or emits a light of a or emits light of a particular wavelength range. The material must consume energy first, and this energy may come from several sources such as luminescence, which defines the subsequent emission. And spectrophlorometry includes molecular luminescence techniques. Okay, so one is absorption, one is emission, and then there's this scattering spectroscopy, which tests the amount of light at specific wavelengths, which also looks at the incident angles and polarization angles that a material in question can scatter. The method of scattering is actually much quicker than the process of absorption or emission. So, Raman spectroscopy is one of the most beneficial applications of light scattering spectroscopy. Now, this absorption spectroscopy, I think we looked at uh, Beer-Lambert's law earlier as well, a little. Uh, I mean, not little, where we used to see the how do you really determine the concentration of the purified plasmid, okay, as well as the purity of the sample, like A260 by 230 ratio, A260 by 280 ratios, those things. So absorption spectroscopy is based on the law known as Beer-Lambert law. Now when an electromagnetic radiation passes or falls onto a homogeneous medium, some amount of light can be reflected, some amount can be actually absorbed, and rest of the radiation can be transmitted by the matter in question. Now, absorption spectroscopy is actually governed by two laws, Lambert's law and Beer's law, which is actually combined together to known as finally giving the term Beer-Lambert law. So what does Lambert's law suggest? It states that when a monochromatic light means whatever the medium, whatever the medium is, basically the monochromator in question, it will allow only uh, out of several wavelengths, it will allow only a certain wavelength of light to pass through it. Okay, so it states that when monochromatic light passes through a transparent medium, the intensity of transmitted light decreases exponentially as the thickness of the absorbing material increases. Right? Okay. So this is what Lambert's law uh, uh, postulates, and there's Beer's law, which states that the intensity of the transmitted monochromatic light decreases exponentially as the concentration of the absorbing substance increases. Okay, so if the concentration of the solution in question, suppose you have a lot of nucleic acid in your, like a lot of plasmids in your purified sample, right? So eventually the concentration that you will get, the transmitted light, so you will have a lot of molecules in the solution to actually absorb the radiation that is shown onto it. Okay, right? I mean, that is passing through the sample. So there are a lot of molecules inside it because the concentration is higher to really absorb the light that is passing through the sample. So the eventually the transmitted light, what is given off finally, uh, is lesser as compared to, suppose a cleaner solution like only water, and you have inside water you have plasmid or plasmid DNA suspended all throughout. So definitely there are these nucleic acids there, DNA molecules there, which will eventually absorb some of the light, and the transmitted light, amount of transmitted light will decrease. Okay. So this is what there is the light source, and you have a monochromatic, which will eventually help in uh, passing only a certain wavelength of light. So this light source can emit light of different wavelengths. Okay, and then this is incident on a sample that is present in a cuvette of a particular thickness. Okay, so based on what is the concentration of the solution inside the cuvette, this is a transparent medium. Okay, and the light can be transmitted. The transmitted light is actually detected, and eventually we get a absorbance value. So basically the absorbance is directly proportional to the thickness of the medium. That's what we looked at, and the concentration of the medium. So when we the proportionality constant is epsilon here, which is the absorption coefficient. Okay. Now, transmittance is another way of describing the absorption of light, right? So, transmittance T is simply the ratio of the intensity of the radiation transmitted through the sample to that of the incident radiation. Now, both these like absorbance and transmittance are actually dimensionless quantities. Okay, so that's why they are usually represented as AU or arbitrary units. Okay. So, now, even in the uh, like UV region, there's infrared region, then there's this visible spectrum. So UV region is mostly nearly 10 nanometers to nearly 400 nanometers long. Then there's near UV as well, where the wavelength ranges between 250 to 400 nanometers. Then there's far UV region as well, which is 190 nanometers to 250 nanometers. Then the visible region we looked at. Then Then there's vacuum UV region as well, where the wavelength is less than 190 nanometers. Now, when a specific wavelength of light hits a molecule, that molecule gets excited, right? So basically what we are talking about excited is the electrons in question. We are talking about the electrons in question. Now, once the electron excites, it excites from the ground state or a lower energy state to the higher energy state, right? Because the electron is eventually absorbing some amount of energy. So it will eventually can, reach up to higher energy levels. Now, when that particular electron actually jumps off, it absorbs light energy because electrons in the orbital at a lower energy state utilize energy to move to a higher energy level. Now, for it to come to a lower energy state, that means it will eventually release off some energy or give off some energy. Okay. So, energy is actually neither created nor destroyed, but it can be transformed from one form to another. Now, on passing an electromagnetic radiation, suppose in the UV visible range of 200 to 800 nanometers, only light possessing this precise amount of energy. So, E is equal to h nu, right? Which E is equal to h nu, nu is frequency, and then h is Planck's constant. So, nu can also be written as c by lambda. Okay. So, I mean the speed of light by the wavelength of light, right? So that's why uh, energy is actually inversely proportional to the wavelength of the light. Or even these two, like frequency and wavelength, are also inversely proportional. Where was I? Huh. Only light possessing this precise amount of energy. So this is what we are talking about. Uh, so this is in wavelength terms. So it can be written as hc by lambda. You can get the energy range of this UV visible range. So this particular range of energy can only uh, cause transitions from one level to another, and these will the matter's energy levels are quantized eventually. Okay, now if the energy is utilized, the intensity of light received is lost, and during this time the energy absorbed by the electrons will equal the energy difference between the energy levels, whatever, like lower and higher energy levels. So if one needs to, like, ground level 0 to level 1, energy level 1 0, if some electron is at ground state, that means it doesn't have any, I mean, let's suppose it doesn't have any energy as such, but then whatever it needs to, needs to have some amount of some x amount greater energy for it to jump into the level 1. Okay, so that uh, x amount of energy is basically the energy difference between the ground state and the first excited state. Okay, or level 1, energy level 1. Okay, so this is what is basically being talked about. The energy absorbed by the electrons will equal the energy difference between the two energy levels, and during this stage electron transition occurs. So when a particular matter or a molecule interacts with an electromagnetic radiation of a certain wavelength or of a certain uh, energy, the spectra received are called absorption spectra. Hence, it is called like spectroscopy. And similarly, when electrons in the orbital at a higher energy level move to the ground level, the spectra received are called emission spectra. Okay. So there is something called single beam spectrophotometer, so where the light enters the instrument in the setup through an entrance slit here, um, is collimated and then focused onto a monochromator, which eventually only passes light of a certain wavelength or the desired wavelength, and then uh, there's your sample as well. And definitely one thing, so you need to have a, suppose when we talk about the density of cells, E. coli cells in a liquid culture medium, so to look at that you need to have the blank LB media as well, like inside which the your E. coli cells are growing. So based on that reference or based on that standard, one will eventually calculate the concentration of the cells. And the intensity of the transmitted light is detected by the detector and eventually converted to absorbance or transmittance. We get the absorbance or transmittance ratios. So there's single beam spectrophotometer here, and there can be double beam spectrophotometer as well. So a double beam spectrophotometer has two light beams, one of which passes through the sample while other passes through a reference cell here. Okay, this allows more reproducible measurements because any fluctuation in the light source or instrument electronics appears in both the reference as well as the sample of the cell, and therefore can easily be removed from the sample spectrum by subtracting the reference spectrum. So, some of the most commonly used detectors in a double beam spectrophotometer, or in general the UV visible spectrophotometers, are PMT or photomultiplier tubes. The monochromator, the one which allows only certain wavelengths of light to pass through. So, the monochromators in these double beam spectrophotometers is placed after the sample so that the sample is exposed to the entire spectrum of the electromagnetic radiation, or the incident radiation, and the transmitted radiation is dispersed into its components. Okay. So PMT, the photomultiplier tube that we were talking about early in the just the previous slide, PMT's function, it actually functions by the photoelectric effect. What happens in photoelectric effect? Like in very simpler terms, it's there are electrons are emitted from the surface of certain types of metals when because they absorb a light packet, or a photon of light energy such as visible or UV light. Now PMTs function by a photon striking the first dianode. Dianode are basically electrodes which are emitting the electrons when those electrons are actually hit by a packet of light energy. Okay. Which releases electrons into a diode chain with progressively higher voltages, and eventually this causes an electron cascade down the electrodes until the electrons hit the anode, which generates the electrical charge that can be finally measured. So the net result is one photon releases many electrons onto the anode. Okay, so this is how the PMT or the photo here it was measured. The detector, this one, the photomultiplier tubes function. Okay, next we come to infrared spectroscopy. So infrared region is considered around this range. The energies associated with molecular vibrations are smaller than those associated with electronic transitions and fall in the IR region. Basically, this is used to probe the vibrations in molecules, and IR spectroscopy is also known as vibrational spectroscopy. Now, again, this particular region is divided into three subdivisions. One is the near infrared, there is the middle infrared, and the far infrared regions. Okay, wave numbers are basically inverse of the wavelength value. Okay, so mostly IR spectroscopists use wave numbers or nu bar to represent the IR spectra, and suppose if mid IR region is in the wave number range of 4000 to 400 centimeter, like inverse centimeter, or wavelength is 2.5 to 25 micrometers, is the region of interest for studying molecular vibrations. Okay, now photon energy is associated with this part of the infrared from 1 to 15 kilocalorie per mole are not really large enough to excite electrons, but these can induce vibrational excitation of covalently bonded atoms and groups. It won't really detach, and the electrons won't really go off to some, like, won't really transition between energy levels, but the covalently bonded atoms or groups of molecules can eventually vibrate. Okay, so that is what is measured by IR spectroscopy. When we talk about degree of freedom and molecular vibration, just these are these are just an overview. No need to like really go really deep inside, just for uh, a basic concept. So in a 3D space, an atom in isolation can have 3 degrees of freedom corresponding to the motion along the three independent coordinate axes xyz. So a molecule composed of n number of atoms has actually a total of 3n degrees of freedom, right? Now when we are talking about uh, if we want to find the degrees of freedom of a diatomic molecule. Okay, so a diatomic molecule can have 3 into 2. Two atoms are there, 6 degrees of freedom. How? So 3 of these 6 degrees of freedom correspond to the translational motions along the uh, axis, XYZ axis. 2 of them define the rotational degrees of motion. One can eventually rotate along this uh, directions, and one corresponds to the vibration of the atoms along the bond. Okay, and we can leave this part. Now I think this is a video. Yeah. Now see the normal modes of vibrations are asymmetric, symmetric, wagging, twisting, scissoring, and rocking for a polyatomic molecule in question. Okay, so symmetric stretching is something like here. It's like really symmetric. Okay, so these are all vibration modes. Asymmetric is like you see there's an asymmetry here. Then there's wagging here, like how a dog wags its tail, right? And then there's twisting here between the molecules in question. Then there's scissoring effect, how you really use the scissors to cut paper. And then there's rocking effect as well. Okay, these are the different modes of vibration uh, that one can have along the bond in question. So, oh, sorry. So the absorption of infrared radiation, a molecular vibration is IR active, that is, it absorbs IR radiation if the vibration results in a change in the dipole moment. Okay, so suppose there's a diatomic molecule that has only one mode of vibration. We looked at it, a diatomic molecule can have 6 degrees of freedom, right? So yeah, out of that only one mode of vibration is there. So that mode of vibration may not absorb an IR radiation if the vibration does not accompany a change in the dipole moment. Okay, so this is actually true for all the homonuclear diatomic molecules like O2, H2, because those basically don't have any difference in their partial charges. Okay, so like H2, N2, O2, these are homonuclear diatomic molecules. There's no change in the dipole moment here. Now, vibration of when we talk about C and O is more electronegative as compared to C, right? So O will have a partial negative charge like delta minus over it as compared to C, which will have delta plus charge on it, right? So vibration of carbon monoxide on the other hand causes a change in the dipole moment. There's partial charges develop over the individual atoms here, and is therefore this heteronuclear diatomic molecule is IR active. Okay, so for any molecule to be IR active, uh, there needs to be vibration definitely, but the vibration should eventually lead to a change in the dipole moment. Okay, so vibration of a bond involving two atoms that have that have large electronegativity difference is usually iron active. This is what is happening here, right? O is more electronegative than, uh, as in O has a higher tendency to pull the electrons towards itself as compared to electron cloud towards itself as compared to the C atom. Okay, so that's what leads to development of the partial positive charges on carbon atom and partial negative charges on the oxygen atom. So an IR active vibration of a particular frequency absorbs the IR radiation of the same frequency. Okay. Now, this is an example to calculate the position of absorption band for carbonyl stretching vibration, CO, where the frequency is given that these many vibrations per second in acetone. Okay, so the CO group, they're asking to basically to calculate the wave number. By position, it means it is asking to calculate the wave number. So wave number is what? Inverse of lambda, and you have the frequency given here. Okay, so basically lambda is what? C, uh, if it's inverse of lambda, then it becomes nu by c and centimeter inverse, and eventually you put the values in there, you have the speed of light here. And also just remember to, because it's centimeter inverse, remember to convert the values, like if it's meter per second, remember to convert it to centimeters per second, because light speed is 3 into 10 to the power 8 meters per second, or 3 into 10 to the power 10 centimeters per second, right? So this just needs to be kept in mind, otherwise the calculations are pretty simple. Okay, now this helps in identifying the functional groups. So IR spectroscopy actually allows identification of functional groups, like suppose a carbonyl group in question, CO group, is an interesting functional group worth discussing. So carbonyl is a double bond with a very high polarity because there's a electronegativity difference between C and O atoms. Now, stretching vibration of carbonyl group causes large changes in the dipole moment, consequently resulting in a very intense absorption band. Okay. Furthermore, the frequency of carbonyl stretching does not differ significantly for aldehydes, ketones, carboxylic acids, and esters. So all these have C double bond O, right, in some form or the other, right? So the large intensity and relatively unchanged frequency of carbonyl stretching in all these different forms of compounds uh, allows easy identification of carbonyl compounds, basically the compounds containing C double bond O group. Uh, and there is one thing to note, that carbonyl stretching frequency can be much lower for amides and much higher for anhydrides and acid chlorides. So all these basically also gets influenced by what are the surrounding atoms as well, like whether they are more electronegative or more electropositive as compared to the atom, like in question, the C double bond, I mean the at molecules in question, C double bond O, what are the neighboring atoms lying around. So eventually, which one pulls the electron cloud more towards itself, the changes in the dipole moment eventually shapes whether the stretching frequency is higher or lower. Okay. So this is again typical vibrational frequencies of different kinds of functional groups. Okay. We looked at C double bond O containing groups like aldehydes, ketones, esters, carboxylic acids. This lies in between this range. So this is just a table for information. This is taken from the lecture video only. Now let's look at some of the questions. So, which part of the electromagnetic spectrum has the longest wavelength? Longest wavelength means it has the least amount of energy or least frequency. So that will be radio waves. Okay. Next, UV visible spectroscopy is primarily used for what? Um, Mostly it's for characterizing the electronic transitions. And another question from UV visible spectroscopy, the absorbance of an analyte is directly proportional to, this is basically from the Beer-Lambert's law. So the absorbance of an analyte is directly proportional to the concentration of the sample in question. The option B, concentration of the analyte. Which type of atomic or molecular processes are typically measured using UV visible spectroscopy? It's more or less the same question. It is electronic transitions. Which spectroscopy method among the following is suitable for identifying functional groups? We just looked at it now. Vibrational spectroscopy. Electron paramagnetic resonance spectrum spectroscopy uses what kind of radiation? So radio frequency region has very low energies because the wavelength is the longest, among the longest in the electromagnetic spectrum. And these correspond to energy differences in the nuclear and electron spin states. And these frequencies therefore find their applications in NMR and electron paramagnetic resonance spectroscopy. So what is EPR? or EPR is also known as electron spin resonance spectroscopy, ESR, for studying materials that have unpaired electrons, right? There are paramagnetic and diamagnetic atoms as well, right? I mean, paramagnetic, diamagnetic have paired spins. Um, the basic concepts of EPR are analogous to those of nuclear magnetic resonance, but the spins excited are those of the electrons instead of the atomic nuclei. So, nuclear magnetic resonance is the spins of the atomic nuclei is taken into consideration, whereas in electron spin resonance, the spins of the excited electrons, there will be electrons here, or those of the electrons are taken into consideration in EPR or ESR. So, sorry. So the previous question, the answer will be electron paramagnetic resonance spectrum spectroscopy uses radio frequency waves. Okay, because these correspond to the energy differences in the nuclear and electron spin states. Okay, so a UV visible spectrophotometer can measure absorbance or optical density. Okay. So there is this hydrodynamic volume fluorescence. I guess everyone will know. There is a term called zeta potential here. This is basically the electric potential at the slipping plane. Basically, the interface which separates a mobile fluid from the fluid that remains attached to the surface from where the fluid is slipping off. Okay. So the zeta potential actually is a measure for colloidal solutions. The zeta potential is an important and readily measurable indicator of the stability of colloidal dispersions. The magnitude of the zeta potential indicates the degree of electrostatic repulsion between adjacent or similarly charged particles in a dispersion. Now, when we are talking about molecules and particles that are really small, a high zeta potential will confer stability. That means the solution or dispersion will resist aggregation, right? Now, when the potential is actually small, attractive forces may exceed this repulsion and the dispersion may break and flocculate. So, colloids with a high zeta potential means negative or positive value, are electrically stabilized, while colloids with low zeta potentials tend to coagulate or flocculate, uh, as outlined in the table. The table, I don't think it's there. Uh, anyway, so that is what is zeta potential. Basically, it is used for measuring the uh, stability of colloidal dispersions. The dash of a spectrophotometer splits light into various wavelengths. Okay, we have been talking about it multiple times. Answer is option C, monochromator. Dash stops the reformation of disulfide bonds. So DTT, BME, all these simply reduce the disulfide bonds. It like introduces thiol groups SH, but iodoacetate is something which leads to um, like permanent modification of the removal of the H attached to the S. Okay. Now, how many degrees of freedom does a diatomic molecule have? It is 3n, which is 3 into 2, 6. Degree of freedom is the number of variables which are required to describe the motion of a particle completely. Okay. Now, when we are talking about an atom moving in a 3D space, there are three coordinates are adequate, like enough to really describe its motion. So, the degree of freedom is 3 here. Now, in this context, its motion is purely translational, that is along the individual axis, x, y, and z axis. Now, if we have a molecule made up of n atoms or n number of ions, now the degree of freedom becomes 3n because each atom now has 3 degrees of freedom. And along with that, since these atoms are bonded together, all motions cannot be translational because the bond location of the bond is along some direction as well. Okay, so the presence of that particular covalent bond will also restrict movement. So all motions are not translational. Some can become rotational along the covalent bond in question. Some can vibrate along the bond. Um, so for non-linear molecules, all rotational motions can be described in terms of rotations around the three axes. The rotational degree of freedom is 3, and the remaining 3n minus 6 degrees of freedom constitute the vibrational motion. And for a linear molecule, however, rotation around its own axis is no rotation because it will leave the molecule unchanged. So whenever we are talking about any kind of rotational degree of freedom, then that means there is some change in the molecule, in the conformation of the molecule. Okay. So if we are talking about a linear molecule, rotation of that particular molecule, it will actually rotate around its own axis. So there is eventually the molecule is finally that we are getting after the rotation along its own axis, it's eventually leaving the molecule unchanged, right? So there's no rotational degree of freedom around its own axis for a linear molecule. So there are only two rotational degrees of freedom for any linear molecule, leaving 3n minus 5 degrees of freedom for vibration. Okay. Now, the position, basically it's asking for the wave number. Position for the absorption band for carbonyl stretching vibration. This is, I think, the same question. This is 10 to the power 13. Okay. So it will eventually be, if it's wave number, it's 1 by lambda. And if it's 1 by lambda, you will eventually have frequency by speed of light. Just make sure you are keeping the units same in the numerator and denominator. So the correct option is option A. I think this is the same question from the example. In an IR spectrum, which region corresponds to the fingerprint region? Now, a fingerprint region is basically the region where a large number of infrared bands can be found, and many different vibrations, including CO, CC, CN single bond stretches, CH bending vibrations, and some bands due to benzene rings are found in this region. The fingerprint region is actually the most complex and confusing region to interpret and is usually the last section of a spectrum to be interpreted. However, basically it is by interpreting the fingerprint region of the infrared band, in, you are getting the composition of the molecule. Okay, you are getting the fingerprint of the molecule. So that's why it's called a fingerprint region. So the stretch is actually this stretch 1500 to 400 centimeters inverse. Okay. Now, this is something uh, some of you might know. Based on the wavelength versus the molar absorption coefficient, what is the composition of the sample, whether it's highly concentrated or less concentrated, and if there are any fluorophore in question or any kind of light chromophore in question. So there can be factors that could shift the absorption spectra to smaller wavelengths towards the left, or it can also increase or decrease the absorption intensity. So so there can be, so these are few terms like bathochromic shift. So basically, if this is the absorption spectrum, it shifts towards longer wavelengths, like towards the right. So then it's a bathochromic shift, or it is also many times called as red shift. Okay, then there's hypsochromic shift of the absorption spectrum towards smaller wavelength. This is also called as blue shift. Then there's hyperchromic shift, where the intensity, molar absorption coefficient or intensity of absorption increases, and hypochromic shift, where the intensity of the absorption will decrease. Okay. So this is what the next questions are about. A hyperchromic shift is increase in the absorption intensity. Now, when an oxochrome is attached to a chromophore, so what will actually happen? Whether there will be a blue shift, red shift, a hyperchromic shift, or a hypochromic shift. So what is actually happening is it will shift to longer wavelengths. Okay. So these oxochromes, It is a group that does not behave as a chromophore on its own, but when it is actually linked to a chromophore, it shifts the absorption. Uh, this will be absorption towards longer wavelengths while increasing the intensity of absorption. So, oxochrome enhances a molecule's color, whereas chromophore imparts the actual color to a molecule. So, oxochromes are basically helping molecules, mainly color enhancing groups. So, there are some examples like OH, NH2, NH, and some group, functional group attached to it, or no N, only two functional groups attached to N. So, these are different groups that can behave as oxochromes. Okay. So, that's it for today's tutorial session. We will meet again the next Saturday. Okay. Stop. Thank you all for joining. I will end the meeting for everyone. Yeah. Yeah. Thank you, Priscilla. Thank you, Dashisha.